Welcome to an overview of Parkinson's disease that moves from basic mechanisms to therapeutic breakthroughs. This slide frames our focus on pathological mechanisms, biomarkers, and neuroimaging, and how integrated research and clinical development guide new treatment horizons. Picture the center Venn diagram as the roadmap linking discovery science to therapeutic innovation and high-density consulting insights. The history of Parkinson's understanding unfolds from James Parkinson's 1817 description to the 1960s discovery of dopamine deficiency and the levodopa revolution, and finally to today's multi-system view. The images trace that evolution: clinical observation, biochemical breakthrough, and the modern recognition of Lewy bodies and genetic factors that expanded Parkinson's concept beyond a single movement disorder. About 10% of cases are monogenic, and this slide highlights how rare inherited mutations illuminate disease biology. Dominant mutations in alpha-synuclein and LRRK2 promote toxic protein aggregation, while recessive losses like PRKN and PINK1 impair mitochondrial quality control. Together, these genetic pointers show how protein handling, mitochondria, and lysosomal function converge to increase dopaminergic neuron vulnerability. This slide explores environmental and lifestyle contributors and their interaction with genetics. Chronic pesticide exposure, rural living, head trauma, and inactivity are linked to higher risk, while a Venn diagram shows how environmental toxins, alpha-synuclein misfolding, and oxidative stress amplify cellular pathology. On the right, modifiable protective associations and public health strategies suggest potential preventive avenues worth exploring further. The alpha-synuclein slide walks through misfolding to aggregation, Lewy body formation, and prion-like transneuronal spread. Visuals show soluble oligomers progressing to insoluble fibrils, and the idea that pathogenic seeds transmit between donor and recipient neurons. Clinically, this framework highlights intervention points where targeting aggregation, propagation, or clearance could slow disease progression. Mitochondrial dysfunction is a core theme here, starting with complex I inhibition that raises ROS and lowers ATP, then moving to PINK1 Parkin-mediated mitophagy failure. Damaged mitochondria accumulate, clearance is impaired, bioenergetic collapse and calcium dysregulation follow, and ultimately dopaminergic cell death occurs. The slide ends by pointing to PGC1-alpha-mediated biogenesis as a promising neuroprotective strategy. This slide links neuroinflammation and the gut-brain axis, showing how alpha-synuclein aggregates and activated microglia release inflammatory mediators like IL-1 and TNF-alpha to drive neurotoxicity. The middle panel depicts intestinal dysbiosis, increasing LPS permeability and systemic cytokines that may ascend to the brain. On the right, modulating the microbiota or blocking inflammasomes appears as a therapeutic attenuation strategy. The clinical progression slide traces the journey from nigral neuron loss to motor complications. Early-stage dopamine loss produces bradykinesia, tremor, and rigidity. Levodopa restores function initially, but over 5 to 10 years, patients often develop wearing-off and peak-dose dyskinesias due to pulsatile stimulation. Practical challenges like erratic gastric emptying and altered dopamine buffering make adjunctive therapies necessary. This slide highlights non-motor features that often define Parkinson's early and late course. Prodromal symptoms include constipation, REM sleep behavior disorder, hyposmia, and mood or executive changes. Mid-stage brings pain, apathy, and early cognitive decline. Late stage can feature severe dementia and quality of life decline. A key point is that non-motor signs often respond poorly to dopaminergic drugs and need tailored management strategies. The current pharmacological landscape still centers on levodopa plus carbidopa as the most effective symptomatic treatment, with MAO-B and COMT inhibitors used to extend levodopa's benefit. dopamine agonists to delay initiation, and agents like amantadine or anticholinergics for selected symptom relief. The important limitation is that none of these approaches meaningfully halt underlying disease progression, and long-term complications remain a challenge. This slide contrasts continuous dopaminergic stimulation with deep brain stimulation as advanced options for motor fluctuations. Intestinal levodopa-carbidopa gel and subcutaneous apomorphine aim to provide steady dopamine levels, reducing off-time and dyskinesia, while deep brain stimulation of the subthalamic nucleus, or globus pallidus, can modulate pathological oscillations and improve quality of life, though patient selection is critical. Emerging neuromodulation and personalized protocols are highlighted here, from closed-loop DBS that adapts stimulation to neural signals to non-invasive deep stimulation methods like focused ultrasound, and neuromodulatory approaches such as vagus nerve stimulation or dual-site RTMS. The slide also emphasizes the shift toward biomarker-guided, personalized protocols entering clinical testing to tailor treatments to patient-specific biology. The clinical pipeline targets core disease mechanisms, including alpha-synuclein and LRRK2-directed therapies with antibodies, antisense oligonucleotides, small molecules and kinase inhibitors, mitochondrial support strategies like ursodeoxycholic acid and other boosters, and gene therapies aimed at restoring dopamine synthesis or providing trophic support. Visuals make clear this is a multi-pronged effort, linking mechanism to intervention. The final slide frames a paradigm shift toward precision neuroprotection by integrating genomics, proteomics, and wearable sensors to stratify patients into molecular subtypes, enabling combination trials that simultaneously target synuclein, mitochondria, and inflammation. Early biomarkers and gut microbiome signatures are presented as tools to detect disease before neuronal loss and to test interventions designed to achieve true neuroprotection rather than only symptom relief.